Okay. So firstly, we'll see about what are proteins. Proteins are nothing but polymers of amino acid joined together by peptide linkage, and the peptide linkage form is like this: C double bond O NH. Clear? Then after this we discuss how peptide linkage is formed. Okay, peptide linkage is formed by reaction between carboxylic acid group of first amino acid and amino group of second amino acid, and they will form a peptide linkage with the removal of water molecule. Water molecule is get removed, and here the peptide linkage like C double bond O NH is going to form. Okay. Then next is the what is exactly peptide linkage. Okay, peptide linkage, when it is formed, if you want to join proteins, number of proteins are there. So in for the joining of the peptide linkage of the proteins, we require a peptide linkage. Okay, consider कर लो कि आप लोगों की 3 amino acid continuously join. Okay, so to join that 3 different amino acid, the 2 linkages will be formed, right? And that linkage is of peptide. Okay, so at that time we will see a dipeptide linkage is formed. Okay, same way, if 4 amino acid are going to join, okay, if 4 different amino acids are there and in between the 3 linkages we require, okay, so at that time it will be the tripeptide linkage. Okay, then we see the properties of the protein. Okay, proteins are insoluble in water, they form viscous solution. Okay, each protein has a characteristic odor. Okay, consider कर लो कि आप लोगों को albumin पता है egg में से, right? That albumin, it is also having a particular odor. Okay, then at isoelectric pH, firstly what is exactly isoelectric pH? Okay, isoelectric pH means a pH condition where the net charge of the solution is zero, and when that net charge of the solution is becomes zero, whenever there is a presence of same number of cations or same number of anions, at that time जो कुछ भी solution का net charge होगा वो that will becomes a zero. Okay, so at this isoelectric pH, proteins have least solubility as well as they are electrically neutral. Proteins can be precipitated by acids, alkalis, as well as various types of metal ions. Okay, and next one, whatever the structure of the protein is there, if in that structure disturbances are observed or disruption, अगर आप लोग कुछ structure disturb कर रहे हो, okay, so that is affect going to the functional ability of protein. Okay, then next is the roles of protein. Okay, what are the exactly roles? We already know proteins are present in almost every part of the cell. Okay, so whatever the enzymes which are responsible for metabolic reactions into the human body, all enzymes are proteinous in nature. Various types of hormones or many hormones are proteins. Just like you consider insulin, it is a type of hormone that is nothing but a protein in proteinous in nature. Okay, so proteins involved in transport of compound from one part of the body to the cell membrane or to the another parts of the cell. Okay, so if we consider this point, at that time we'll take an example of hemoglobin. Hemoglobin, what it does, he will transport pick or he will transport oxygen from lungs to the other body tissues. Okay, then some clotting factors, clotting के लिए क्या important रहता है, fibrinogen. Okay, that fibrinogen, it is also basically proteinous in nature. Okay, so that's why some clotting factors, they are also proteinous proteins in nature. Then protein confers immunity against a viral and bacterial infection, because whatever the immunoglobulins and antibodies that are responsible for the immunity, that basically are proteinous in the nature. Okay. Then proteins are act as buffer to maintain the pH of the cell, because if we consider about the blood, okay, in blood there is a presence of plasma. This plasma, it is responsible to maintain the pH of this blood, okay, and this plasma is basically containing, or it is proteinous in nature. Okay, proteins are constituent of the blood. Some proteins are having a specific function, okay, and they will provide energy to the body when they are taken in the diet. Then we will see about the classification of protein. Protein mainly classified in the four ways. First, based on the function. Second, based on the molecular shape. Third one is based on the physical and chemical properties. And fourth one is the based on the nutritional states. Okay. So we'll see firstly about based on the function. In that, it is again subclassified in almost 8 types. Okay, in that 8 types, contractile protein is there first. Okay, contractile proteins are responsible for contraction of the skeletal muscle, and the example of these proteins are actin and myosin. Okay, actin and myosins are located in the skeletal muscle, and they are responsible for the producing contraction in the skeletal muscle. Okay, then second is the buffer protein. Buffer protein, buffer we already know, which is responsible to maintaining the pH of the cell. Okay, so buffer protein, it will maintain the pH. Okay, and for this example is the plasma protein, and we already discussed that plasma, it is present in the blood. Okay, then third one is the storage protein. Okay, so storage protein का example है myoglobin. Myoglobin, it is mainly responsible to store oxygen in the muscular tissues. Okay, that's why myoglobin is known as a storage protein. Then next is the structural protein. Structural protein, it is mainly working as giving a certain shape. It will give a certain protective layer or protective coating. The example is the keratin. Keratin, it is generally present in hairs as well as nails. Okay, so both are whatever the keratin which is present in the nails and hairs, it will give a protective coating, as well as it will responsible for the giving a particular shape to the that part. Okay, then next is the transport protein. Example is the hemoglobin. Hemoglobin, it is involved in the transportation of the oxygen from lungs to the tissues. Okay, then next is the regulatory protein. Regulatory protein, in that some proteins are there that are responsible to maintain or to regulate certain metabolic processes. Like consider the example of insulin. Okay, insulin is responsible to maintain the blood glucose level. Whatever we eat a food, from that some uh, from that food material, whatever the carbohydrate part or sugar part is there, that will enters into the blood circulation, and in this way the blood sugar level is maintained. Okay, or blood sugar level is going to follow. This blood sugar level, it is regulated or it is controlled by the hormone known as a insulin. That's why insulin, it is also known as a regulatory protein. Then next is the enzyme proteins. Whatever the enzymes are there, just we discuss in the roles. All enzymes are proteins in nature. Not only glucokinase, we can consider hexokinase, we can consider phosphofructokinase. All the types of enzymes are proteinous in nature. Then next is the defense protein. Example, I have given only immunoglobulins, but antibodies, they are also proteins, and both are responsible for the immunity. And why immunity is required? Immunity is required to defense or to fight against the bacterial and viral infection. Okay, that's why immunoglobulins as well as antibodies, different present in the human body, they are known as a defense protein. Clear? Then second one is the based on the molecular shape. Whatever the proteins which are based on the molecular shape, they are subdivided into two different types. First one, fibrous protein, and second one, globular protein. Okay, fibrous and globular, the name itself indicates fibers means just a fiber-like structure is observed, and globular, it will observe in this spherical shape. Whatever the fibrous proteins are there, these fibrous proteins, they are generally insoluble in the water, and whatever the globular proteins are there, they are soluble in the water. Fibrous proteins, they have a high molecular weight, whereas globular proteins, they have a low molecular weight. दोनों का comparison करके बोल रही हूँ, that's why it will understand easily. Fibrous in shape, globular spherical in shape. Fibrous proteins are insoluble in water, globular proteins are soluble in water. Fibrous proteins are high molecular weight substances, whereas globular proteins, they are low molecular weight substances. Fibrous protein का structure आप लोग consider करते हो hairs, okay? Hairs में जो भी keratin present होता है, that is fibrous in nature, okay? Whereas globular protein, if we consider, at that time we will take an example of albumin, which is present in the egg, okay? So that is the example of the globular protein. Then next is the third classification में third one, that is the based on the physical and chemical properties. Okay, based on physical and chemical properties, it is sub-classified in the three types. First one is the simple protein, second one is the conjugated protein, and third one, that is the derived protein. We'll see about the first one, that is simple protein. The simple proteins, they are defined as those proteins which upon hydrolysis will give amino acids or derivatives of amino acids. Okay, these type of proteins are known as a simple protein. Okay, simple proteins are again having certain example like albumins, globulins, glutenins, and prolamins. Okay, so albumin, globulin, glutenins, and prolamins, they sometimes, for this, if we discuss about this solubility of the compounds as well as heat coagulation property, at that time albumins and glutenins, they are water soluble. Okay, whereas globulins and albumin and prolamine, it is water soluble, whereas globulins and glutenins, they are water insoluble. And then second one is the conjugated protein. Okay, conjugated protein means conjugation होता है वहाँ पे. Okay, proteins are combined with certain prosthetic group. Prosthetic group means प्रोटीन के अलावा भी दूसरा एक ग्रुप अटैच होता है, एडिशनल ग्रुप अटैच होता है. दैट एडिशनल ग्रुप इज नोन एज अ प्रोस्थेटिक ग्रुप. क्लियर? So conjugated proteins are again subdivided into four types. First one, nucleoprotein, second one, chromoprotein, third one, glycoprotein, and fifth one is the lipoprotein. If you see the names, you will get clear idea what is it exactly, what it contains. Okay, nucleoproteins, it mainly contain nucleic acid. जो कुछ भी RNA, DNA, nucleic acid से, if that are combined with the proteins, they are known as a nucleoprotein. Okay. Second one is the chromoprotein. Chromoproteins, in that it will containing the chromophores. Actually, chromophore basically they are colored substances which is having a particular color. Okay. So these chromoproteins, it is made up of protein is combined with the certain colored prosthetic group that is known as a chromophore. Okay. Third one is the glycoprotein. Glycoprotein, in this glyco word आता है, तो in that, in our mind it is clear that it is nothing but a carbohydrate. Okay, so carbohydrates when combined with the proteins, it will form a compound known as a glycoprotein. Okay, last one is the lipoprotein. Lipo means lipids. Okay, so when lipid substance like fats, oil, waxes, okay, when it will combine with the proteins, they are known as a lipoproteins. Clear? Then third one is the subtype of the physical and chemical properties is derived protein. Derived protein, actually These are formed by partial hydrolysis of high molecular weight proteins. Okay, derived proteins are generally described in two ways: primary and secondary. Okay, then next one is the last fourth type of the protein or fourth class, that is based on the nutritional status. Okay, yeah, on the basis of nutritional status they are classified in only two types: complete and incomplete protein. Complete protein, it will provide all the essential amino acid after digestion. That type of protein is known as complete protein. Then second one is incomplete protein. Those proteins are not going to provide all the essential amino acid after digestion which are required for the body. They are known as a incomplete protein. Okay, this much is about the an introduction to the proteins. Now we'll see exactly protein किससे बनते हैं. They are made up of amino acids, right? So we'll see about amino acid first. Okay, amino acids are structural and functional unit of protein. They are containing amino group as well as carboxylic acid group. Okay, and both are attached to the alpha carbon. Okay, C C double bond OH, that is carboxylic acid group, and NH2, that is amino group. Okay, now we'll see about the classification of amino acids. Amino acids are mainly classified in two ways: based on the nutritional status, and second one is based on the nature of the solution as well as structure of the amino acid. Okay, now we'll see firstly about the based on the nutritional status. Okay, they are subclassified as essential amino acid and non-essential amino acid. Essential amino acids, those amino acids which are not synthesized in the body, but they are needed for certain metabolic condition or certain metabolic reaction. Hence, we have to take that particular amino acid from the diet. Such type of amino acids are known as essential amino acids. Clear? Then next one is the non-essential amino acid. Second, on the basis of nutritional status, that is non-essential amino acid. Non-essential amino acids are synthesized in the body, not hence not needed to be supplied in the diet. Already वो synthesize ही होते हैं, तो whatever the requirement for the body is there, that will get nullified in the body only. Okay, no need to take from the outside diet. Okay, so this type of amino acid particularly, they are known as the non-essential amino acids. Now we'll see about the second classification type, that is known as the based on the nature of solution and structures. Okay, in that first one is acidic amino acid. Acidic amino acid will have one amino group in the structure, and it will containing one amino group in the structure and two carboxylic groups at the pH 6. Okay, so the example of this acidic amino acid is aspartic acid. Okay, this aspartic acid containing the structure in this C double bond OH as well as COOH. Two carboxylic groups are attached with the alpha carbon along with one amino group. Then second one is the basic amino acid. Basic amino acid containing two amino group and one carboxylic group at the pH 6. Okay, see the structure of the lysine. The example of basic amino group is lysine. Okay, then neutral amino acid. Neutral amino acid, it will contain one amino group and one carboxylic acid group. Example is the glycine. In the structure you can easily observe You will easily observe this thing. Okay. Then next is the sulfur containing amino acids. Okay. Sulfur containing amino acids, it will, in the structure there is mainly presence of the sulfur. Okay. See these two examples, methionine and cysteine. Okay. methionine के structure में भी sulfur group है, as well as cysteine के structure में भी sulfur group है. Remember one thing, if any amino acid or if any protein contain a sulfur in the structure, at that time it will form a disulfide linkage. Okay. A disulfide linkage is present, or sulfide linkage is present along with the peptide linkage. Okay. Then aliphatic amino acid, example है serine. Aliphatic means हम लोग बोलते हैं कि a straight chain is there. Okay. So this is the example of serine. Then next is the aromatic amino acid. Aromatic name itself indicates aromatic ring होगी. Aromatic ring अगर आता है तो in our mind it is clear that it is a benzene ring. Okay. So see these two examples, phenylalanine and tyrosine. Both are containing the benzene ring. It is attached with the alpha amino acid. Okay. Only R group it changed. Remaining structure will be the same one. Okay. Then last one is the heterocyclic amino acid. Example is the tryptophan. Heterocyclic means जो कुछ भी benzene ring है, to that benzene ring अगर कुछ attachment होता है, this structure it is of indole ring. Okay, the indole ring it is a heterocyclic ring. Okay, this is attached with the alpha carbon atom, or sorry, alpha amino acid, and it will become a heterocyclic amino acid, and this structure is of tryptophan. Okay. Now we'll move to the next part of the amino acid, about the properties of amino acid. In that first property we will discuss about the zwitterion of amino acid. Basically see the structure of the amino acid. Okay. This is the basic structure of amino acid, in which acidic group is also there and basic group is also there. Okay. So whatever this acidic group and basic groups they are present, they are responsible for the change in the certain changes produced in the your structure. Okay. Why these changes produced? Because what happened, amino acid basically it contain two ionizable group. One is your carboxylic group जो कि acidic group है, and amino group जो कि basic group. Okay. अगर हम लोग alkaline pH maintain करते हो, alkaline pH means pH more than 7. Okay. In alkaline pH, amino acid it will act, or it is present as a anion, means this thing. Okay. C double bond O O. Okay. And if acidic pH अगर हम लोग maintain करते हैं, at that time it will act on the amino group and it becomes a NH3. Okay. So at certain pH, okay, amino acid carry positive as well as negative charge. This formed compound is known as a zwitterion. Okay. Positive as well as negative charge भी है यहाँ पे, NH3 as well as NH3 plus as well as COO minus. This is nothing but here zwitterion. Okay. Then next we'll discuss about the isoelectric pH of amino acid. Basically what is isoelectric pH, or what is exactly isoelectric point? Okay. Isoelectric pH, it is nothing but, or it is the pH when net charge of the solution is 0. And why it becomes 0? Because at this pH condition, or at this point, the number of presence of positive ions as well as negative ions, or positive charge as well as negative charge are same. Okay. That's why the net charge of this solution it becomes a 0. Consider कर लो कि आप लोगों ने कुछ solution लिया है and you have dipped a electrode. Okay. Anode भी और cathode भी डाल रहे हो. Okay. still after passing the current, if no charge is observed. Why no charge is observed, or why movement of the charges are not observed? Because there is the presence of same number of positive as well as same number of negative charges. Okay, that's why no movement of the charges you will observe to the cathode as well as anode. Okay, so at this point your solution shows a net charge 0 because no movement of positive as well as negative ions happens. Okay, that's why in property we study that at isoelectric pH they are least soluble as well as electrically neutral. Why it is neutral now it is clear, because movement ही नहीं होगा उसका, same number of positive as well as negative charges है, तो no movement is observed. That's why it is known as a electrically neutral. Generally when it is observed, because zwitterion, see this zwitterion structure, in zwitterion there is the presence of positive as well as negative charge. Okay, at this pH or at this point it is exists at a isoelectric pH, it does not move in the electric field, it is going to show a net charge of the solution is 0. Along with that, it is containing both positive as well as negative charge to this solution. Clear? Isoelectric pH. See this diagram structure. See this structure. Okay, this is nothing but a zwitterion. If we add a basic media, so at that time it will observe like this, and if we add acidic media low pH, so it will observe like this. Okay, now next we'll see about the amphoteric nature of the amino acid. Basically we know what is exactly amphoteric nature. Okay, amphoteric nature means जो सब्सटेंस एसिड भी बिहेव करता है एंड बेस भी बिहेव करता है, डिपेंड ऑन द एनवायरनमेंट. ओके, इट इज नॉट लाइक दैट कि एनी सब्सटेंस है वो एसिड ही रहेगा, ऐसा नहीं है, एसिडिक ही बिहेव करेगा ऐसा नहीं. सम सब्सटेंस आर जस्ट बेसिक प्रॉपर्टी. ओके, सो in between that some substances are there like your water. Okay, water is the best example of the amphoteric substance. Okay, so amphoteric substances are the substances which act as acid as well as base under the certain different pH condition. Okay, so these substances are known as a amphoteric substances. Normally what happen, see the center amino acid जो भी observe दिया गया है, that is the structure of zwitterion. It contain NH3 plus COO minus. Okay, this zwitterion is actually zwitterion it exists at isoelectric pH. We studied this part previously, right? So this exists at a isoelectric pH, and due to this, if any pH change is occur, that leads to or that may changes the behavior of the amino acid. कंसिडर कर लो कि आप लोग zwitterion ले रहे हो या अमाइनो एसिड ले रहे हो. ओके, इफ दैट अमाइनो एसिड is comes in contact with the acid. Okay, if zwitterion or amino acid comes in contact with the acid, or if we introduced into the solution containing the less pH. Okay, so at that time what it does, it will accept a proton. Okay, it will accept a proton and it is going to convert as a positively charged, and in this way it will act as a base. See, and see in the left hand side. Okay, it will accept a proton and in this way it will going to charge positively, and in this way amino acid act as a base. And in 12th we study that those substances which accept a proton they are bases, and those substances which donate a proton they are acids. Like, so this concept will apply here. So those substances, if amino acid comes in contact with the acidic environment, okay, So acidic environment में contact में आने के बाद it will accept a proton, and after accepting a proton it becomes a positively charged. Okay, positively charged होने के बाद it will act as a base. Clear? Exactly opposite to this reaction. Okay, consider a amino acid when it comes in contact with the basic environment. Okay, basic environment means pH more than the 7. Okay, so at this time it will donate a proton. Okay, proton donate करने के बाद what will happen? It will becomes a negatively charged compound. Okay, so that's why it becomes a negatively charged compound. It will acting as an acid. Okay, it is donating a proton, that's why it is acting as a acid. Clear? Clear?